Assuming you have just created and configured a new xGDBvm instance, you'll now want to test it with sample data to verify that the VM was set up correctly, and also to familiarize yourself with the processes and workflows that you'll follow when you use your own data. First, go to the Manage section you just password protected. You can use the Click to Get Started button or choose from a complete menu of options on the left. Choose Configure Create a Genome Database. The Data Volumes page is a useful first page to visit to verify that external volumes are mounted and have adequate storage capacity. A common problem is insufficient storage space for outputs, which can consume up to 10 times the aggregate size of your input data. Click Learn More or visit the help pages for more details. Next is License Keys. License keys are required for some software installed on xGDBvm, including Genome Threader for protein spliced alignment, vMatch for repeat masking, and GeneMark for ab initio gene finding. The licenses are simple to obtain and free for non-commercial users. Once obtained, xGDBvm makes it simple to upload and install them on your VM. Visit the links shown here to obtain and download license files to your local computer. Then access iPlant's Data Tools site to transfer the uploads to your data store, which you've previously mounted to your VM. Your web browser should allow you to log in without providing credentials again. Under your user home directory is a directory called Keys that was created during the configuration process. This is the destination directory for the license keys, so click to select it. If you had previously uploaded any keys, they would be listed here. Now click Simple Upload in the upper left and locate the license keys one by one on your local drive. Then click Upload to import them to your data store. Click Refresh to confirm that the files have all uploaded, and then return to the license keys page of your VM configuration and refresh the page. There should now be an Install button for each of the 3 programs. Click these one by one to install the license keys on your VM, and note the confirmation checkmark. Also be aware that in future your license keys will again be available to install whenever you mount your data store to a new instance, so you will only need to repeat the final installation step. You are now ready to test your VM with some sample data. Navigate to the page titled Configure New GDB in the left menu. Click the large red button to display sample data sets available for loading. We'll select Example 4, which demonstrates the spliced alignment of 4 data types, along with pre-computed gene models and de novo gene structure annotation using cpGAT. Click to save Example 4, and note that it now displays development status, meaning that no data have yet been processed. Next, examine the validation drop-downs that appear. Under Input, we see that several files have been validated and their data type displayed in green text. xGDBvm uses naming conventions to identify different data types, such as est.fa and gdna.fa. Any file not correctly named will not appear in this list, so you'll want to make a thorough check. You will also want to validate file contents using the eye icon. Click it to view information relevant to that sequence type. Errors such as incorrect FASTA headers or duplicate IDs will also be flagged. Other accessory files, such as reference proteins and repeat mask libraries, if specified, will be listed here as well. Finally, check the expected output, which lists all the feature tracks that will be displayed in the genome browser based on the valid inputs provided. The list will also flag any omissions, including dependent files. You should now be ready to initiate the automated xGDBvm workflow, so click Data Process Options to enter Options mode, and then click Create New Database to start the process. Notice on screen refresh the status is now locked until the process completes. All workflow steps and outcomes are logged, and the most recent step is displayed here. Click to refresh the display periodically to view progress. You can also open the log file by clicking here to view the detailed progress. The cpGAT gene prediction software will take several minutes in this example, as it processes 1 genome segment at a time, a total of 4 in this small example. When the automated workflow finishes, the status will update to current along with a new color scheme. The new genome now also appears as a menu item under the View tab. The configuration page also updates with information about the track data that have been generated, and you can check the totals for each color-coded feature track type. You can also click the icon to view output in a tabular form. For example, red corresponds to EST spliced alignments. The table includes links to genome context views and individual feature records, and it also offers search and other filters to let you focus in on particular data. The same tabular output is available for gene model track features, in this case the blue icon. For this feature type, we also provide coverage and quality scores for gene models that overlap evidence alignments. The same tabular output is provided for CpGAT computed gene structures. You can also view the overall output data in tabular form. The completed log file is also accessible, as is the download page where you can review outputs. Finally, we'll visit the GDB homepage and view the feature tracks in genome context. Our configuration file included a default region that is accessed by clicking this button. Each track is labeled and color-coded by type. Review the alignments, gene models, and masked regions to verify that they are as expected. To recap our second video, testing your VM, first, verify storage capacity, install license keys, configure a new GDB using example data, run the example workflow, and verify output. If all went well, you should be ready to annotate your own genome sequence. See video 3 for a complete description of this process.